Okay, let's dive right into this explainer. We're looking at the incredible, mostly hidden mechanics of photosynthesis and how life on Earth manages to adapt to survive some truly extreme temperatures. You know, we often think of photosynthesis as this super simple static equation, right? Plants take in sunlight and carbon dioxide, and they give off oxygen. Done. But the reality is actually far more dynamic than that. It is a continuous, high-stakes evolutionary arms race against heat, drought, and a rapidly changing climate. I want you to take a deep breath right now. Hold it for a second and exhale. Now imagine taking 9 more of those. Because here's a staggering fact for you. The oxygen in 1 out of every 10 breaths you take is generated by a newly discovered microscopic cellular mechanism that scientists are literally just beginning to understand. It's wild.
So that begs the question, where exactly is all this massive amount of oxygen coming from? Well, we're going to answer that by zooming way, way down to the cellular level of microscopic marine algae. We'll uncover how a tiny, almost mechanical molecular machine supercharges their ability to produce oxygen. And after that, we'll head on to land to see how complex plants have evolved radically different internal architectures just to deal with a warming planet.
Section 1. The Hidden Ocean Oxygen Pump.
All right, our journey starts out in the ocean, specifically with a groundbreaking discovery by researchers over at the Scripps Institution of Oceanography. They were looking at marine phytoplankton, those microscopic organisms that drift around and form the entire base of the oceanic food web. Now, notice how scientists view this VHA enzyme. It's a proton pump, but they think of it like a biological Lego block. VHA stands for vacuolar H+ ATPase, but on its own, its basic job is super simple. Just modify the pH level of its immediate surroundings. But depending on what other proteins it pairs with, this single Lego block can achieve vastly different biological functions. And this brilliantly illustrates just how wild biology can be. That exact same protein pump is used for completely different things across different species. In our own human kidneys, it regulates blood and urine. In giant clams, it secretes a potent acid that dissolves solid coral reefs so the clam can bore a safe little shelter. Deep-sea Osedax worms use it to literally melt the bones of dead whales for food. It regulates blood chemistry in shark gills, and in fish eyes, it delivers extra oxygen to sharpen their vision. I mean, nature is incredibly resourceful. And all of this is before we even consider what it does in oceanic algae.
Because here's what's truly astonishing. In a specific group of phytoplankton known as diatoms, this VHA mechanism acts as an absolute supercharger for photosynthesis. Researchers use these high-tech, super-resolution confocal microscopes and actually found this proton pump tightly wrapping around the diatom's chloroplasts. It pumps protons in to concentrate carbon dioxide, basically force-feeding the chloroplast exactly what it needs to produce massive amounts of sugars. And as a byproduct, it releases a huge wave of oxygen. This single microscopic cellular workaround accounts for up to 12% of the oxygen on our entire planet. Section 2. An evolutionary cellular merger.
So, how did diatoms get this secret biological weapon while other algae completely missed out? Well, we have to look back at an evolutionary accident, a process called symbiogenesis. Looking at the timeline here, about 250 million years ago, a protozoan predator swallowed an algae cell whole. Normally, that predator would just use its VHA proton pump to create an acidic stomach environment and digest its meal. But something went wrong. Actually, no, something went incredibly right. The cell wasn't digested. Instead, the acid from the predator's pump ended up artificially concentrating carbon and accidentally boosting photosynthesis in the prey. Over millions of years, these two completely separate organisms just permanently fused into one, creating the modern diatom. This merger caused a massive, world-altering spike in atmospheric oxygen. And the crazy irony here, the biomass of those trillions of ancient diatoms eventually sank to the ocean floor, and over millennia, transformed into the very fossil fuels we are burning today.
Section 3. Land plants. Spatial solutions. So, while ocean diatoms were busy using a proton pump to concentrate carbon, land plants were dealing with a whole different set of problems. They had to evolve completely different internal anatomies to handle the harsh realities of heat and drought. We can see a really beautiful contrast here between the standard C3 pathway, which is used by about 90% of plants, against the much more highly specialized C4 and CAM pathways. In a standard C3 plant, like rice or wheat, photosynthesis happens basically in one general area. But there is a massive, glaring flaw in the C3 system. The key enzyme that captures carbon dioxide, it's called rubisco, is honestly kind of sloppy. If it gets too hot out, the plant closes its pores, its stomata, right? It does this to conserve water. But that causes CO2 levels to plummet inside the leaf while oxygen levels rise. When this happens, Rubisco gets confused and accidentally grabs oxygen instead of carbon dioxide. This leads to a process called photorespiration, which is basically a massive wasteful drain on the plant's energy. To get around this, C4 plants essentially built a microscopic isolation chamber inside their leaves. Think of crops like maize and sugarcane. They evolved what scientists call Cran's anatomy. Basically, they physically separate the stages of photosynthesis into totally different cells. First, they capture carbon in the outer mesophyll cells and turn it into a 4-carbon acid. Then, they actively ship this acid deep inside the leaf to isolated bundle sheath cells, which is where Rubisco is safely waiting. By pumping concentrated carbon directly into this little VIP isolation chamber, C4 plants completely protect Rubisco from the oxygen, keeping the whole factory running perfectly even when it's blisteringly hot outside.
Section 4. The night shift, cam plants.
But what if you're a plant living in an environment so extreme, like a deep desert where the air is simply too dry to open your stomata during the day at all? You can't just separate your cells spatially. You dry out instantly. You have to separate your entire process by the time of day. This is such a brilliant evolutionary workaround. They solve the exact same problem, but in a totally divergent way. C4 changes where the chemistry happens through spatial separation, while CAM changes when it happens through temporal separation. Crasulian acid metabolism, or CAM for short, is the go-to strategy for about 10% of plant species to survive in places where water is practically non-existent. So the big takeaway here is that CAM plants, we're talking your succulents, cacti, pineapples, they literally work the night shift. During phase 1, at night, when the desert air finally cools down, they open their stomata to drink in carbon dioxide. They immediately convert the CO2 into malic acid and stockpile it inside these large cellular storage tanks called vacuoles. Then phase 3 hits, the scorching sun comes up, and they lock their stomata tightly shut. No water is getting out. They spend their daylight hours breaking down that stored malic acid, feeding a continuous, highly concentrated stream of CO2 straight to Rubisco. They are completely safely photosynthesizing behind closed doors.
Section 5. Adapting to a warming world.
Now, you might be wondering, why does all this matter to us? Well, understanding these intricate pathways from the proton pumps in ocean diatoms to those specialized isolation chambers of C4 and CAM plants isn't just cool biological trivia. It is absolutely crucial for the future of global agriculture as climate change pushes our temperatures higher. Look at how C4 maize and C3 sunflowers respond under drought stress and elevated carbon dioxide. It's really complex dynamics. Elevated CO2 actually reduces the rate of transpiration, which is water loss, in both C3 and C4 crops. Because the air is richer in carbon, these plants don't have to open their stomata as wide to get what they need, so they naturally retain more internal water. That helps conserve soil moisture, which essentially buys these crops a bit of a lifeline during severe droughts.
But here's the profound irony of it all. You would naturally assume that C4 plants, since they evolved in hot, arid regions, would be the absolute, undisputed champions of a warming planet, right? But the data actually shows something super counterintuitive. While C4 plants definitely naturally operate at a higher initial temperature optimum, they have incredibly low plasticity. What that means is they really struggle to adjust their biological machinery when the baseline temperature shifts. Standard C3 plants, on the other hand, possess a way higher ability to acclimate. Especially when we look at woody evergreens and perennial herbs, C3 plants can actively shift their optimum photosynthetic rate to match new changing growth temperatures over time. So, C4 plants might be way less efficient during sudden extreme heat spikes, but they are far more adaptable to long-term, gradual climate shifts.
I'm going to leave you with this thought today. Dr. Tresgears notes that uncovering how diatoms utilize these proton pumps is going to have massive impacts on future research worldwide. As we map out how an ancient 250-million-year-old accident shaped our oceans, and as we deeply understand how C3, C4, and CAM plants navigate extreme heat and water stress, a really incredible possibility emerges. Could these deeply buried cellular secrets, these biological Lego blocks we've been talking about, hold the actual key to engineering the climate-resilient supercrops of our future? It's definitely something to think about.